Hello, and thank you for joining us for this virtual poster session. I'm Shariq Mujib, a product development scientist at Fluidigm, and today I'd like to take you through our poster titled Expanding the Capabilities of the Maxpar Direct Immune Profiling Assay for CyTOF Systems with Additional Markers and Customization of the Analysis Model in Maxpar Pathsetter. So I'd like to give you a brief outline of our poster and invite you to take a closer look at it when you get a chance. I'd like to introduce you to the Maxpar Direct Immune Profiling System and the workflow it entails, and then go into the materials and methods and the results for a small scientific validation study we did with verifying that the additional markers we add, as I'll discuss, worked physiologically accurately, and then we'll discuss the conclusions. So I'd like to introduce you to CyTOF technology, which stands for cytometry by time of flight, which is a mass cytometry technology which uses metal-tagged antibodies for highly multiplex profiling of single cells. It's important to note that each of these antibodies is conjugated with a distinct isotopic mass, which means that it does not suffer from the same signal spillover issues as experienced by fluorescence cytometry and does not require compensation. Therefore, we can design panels comprising greater than 50 markers for comprehensive and in-depth immune profiling studies. In this particular poster, I'll be taking you through a 44 marker panel that we built around the Maxpar Direct Immune Profiling Assay. What is the Maxpar Direct Immune Profiling Assay? It is a ready-to-use dry format of 30 pre-titrated markers that are provided in a single tube for immunophenotyping of human whole blood or PBMCs. As shown in the figure on the top, the process is very straightforward. A user simply adds the specimen into the tube, stains it according to standard staining protocols, acquires it on a mass cytometer, and then can upload that data into the paired Maxpar Pathsetter software, which will automatically analyze the data and cluster into immune cell populations and cell subsets of interest. This makes the Maxpar Direct Immune Profiling Assay ideal for longitudinal studies, especially in the context of studying immune responses in infectious diseases, and has actually already been applied for COVID-19 research and several other infectious disease models. So these are the 30 markers that are included in the Maxpar Direct Immune Profiling Assay. These 30 markers include common phenotyping markers for T cells, B cells, monocytes, and granulocytes in human whole blood or PBMCs, and actually also include markers for further subsetting of these immune cell types into their smaller subsets. Importantly, as also shown in this table, there are 14 additional channels available for users to customize this panel and add in markers of interest to them to really come up with comprehensive panels to study the immune system. I'd also like to tell you that the 14 open channels that were shown in the previous table exhibit minimal signal spillover into in a sample stained with the 30 markers, really suggesting that a user, when they add a marker in here, can result in the generation of high quality data, which will allow for high resolution analysis of their data as well. In this particular poster, we demonstrate that we added 14 antibodies as tabulated over here. These 14 antibodies that we added included high-value functional targets such as PD-1, TIM3, TIGIT, amongst others. And we also incorporated phenotypic markers such as for myeloid-derived suppressor cells, the markers being CD11b and CD33, amongst the other markers shown in the table over here. These antibodies can be obtained from the Fluidigm catalog of reagents or may be custom conjugated using our labeling kits or even using the Fluidigm provided in-house service for custom conjugation. I can show this to you in the form of a wheel. This is the 30 marker panel over here highlighting the open spots as the blank white boxes. And in this particular case, what we did is we filled this in with these 14 antibodies which are now shown in the wheel. So we made a comprehensive 44 marker immune phenotyping panel, which was then tested amongst these samples. So to talk about the samples, I'd like to discuss the materials and methods. So what we did in this particular experiment is that we had PBMC samples from 3 healthy donors and 3 donors diagnosed with rheumatoid arthritis. And what we did is we either rested the PBMC cells overnight or PHA stimulated them overnight. The following day, the cells were stained according to the staining procedures outlined in the Maxpar Direct Immune Profiling Assay. These samples were then acquired on a Helios mass cytometer, which results in the output of an FCS file. That FCS file was then uploaded into the Maxpar Pathsetter software. And in this particular case, we actually modeled updated and modified the Maxpar Pathsetter software model to really highlight the expression patterns of the 44 marker panel. I'd like to showcase what Maxpar Pathsetter software does. So here we're showing a Cen-se plot, which is actually a dimensionality reduction tool, which can take the markers built into the Maxpar Direct Immune Profiling Assay and cluster it into immune cells and their cell subsets. As you can see, each of these clusters can be into the CD4 T cells, CD8 T cells, myeloid cells, B cells, and their smaller subsets, such as their memory phenotypes as well. On the left, we're looking at the 30 marker clustering, and on the right, we're looking at the clustering with the 44 markers. And what's important to note in the dashed circle over there is that by the addition of the extra markers for further stratification of the myeloid cell clusters into the MDSCs, or the myeloid-derived suppressor cells, we can get greater resolution amongst the cluster to further resolve these populations. We can also look at Cen-se plots as a neat tool to visualize the expression patterns of each of these markers on the immune cell phenotypes, on the immune cells that we're interested in. And here we're looking at, from the top left, OX40 to TIGIT expression on unstimulated or PHA-stimulated cells, as showcased over here. What I'd like to direct you to here is that when we look at the expression pattern of each of these functional markers, what's important to note is that the expression patterns align with what is known biologically and is physiologically accurate. For example, we can look at ICOS expression, which is centered around the T cell clusters, but not expressed on B cells. Or we can look at TIGIT expression, which is showcased in particularly memory T cells, but not even within the naive T cells, indicating that the staining patterns we're observing are in line with what is known scientifically. We can also then look at the expression patterns of the phenotypic markers that we added into this panel. And again, we can validate that these expression patterns align with what is biologically known. For example, CD33 expression is observed in the myeloid clusters, and CD2 expression, for example, is shown in the T and NK cell clusters, but not on the myeloid cells, suggesting that we're getting specific staining of the immune cells that we should be getting. To move on to the results, in the first case, we're just looking at the expression of 5 of these functional markers on the total CD4 T cell subsets as identified by their memory phenotypes, divided into naive, central memory, effector memory, or terminally differentiated effector T cells. What I'd like to highlight to you here is that within the healthy donors and the rheumatoid arthritis samples, designated as RA, we observe a trend to increased OX40 expression on the CD4 T cell subsets, which is something that has been published in the literature as well. So we are getting a correct result over here. And then we can also visualize the expression of several other markers on the CD4 T cell subsets. We can also then look at the expression patterns on the unstimulated versus the PHA-stimulated cells. And if I direct you to figure 6C over here, we're looking at expression of PD-L1 and OX40 within CD4 T cell subsets, and observe that within the RA samples, the RA sample CD4 T cells actually exhibit a reduced capacity to upregulate these two functional markers upon PHA stimulation, suggesting that these cells, as has already been noted in the literature, are already immune activated and therefore cannot increase or upregulate the expression of these markers to such a high level. I'd like to also showcase that what Maxpar Pathsetter does is it actually automatically analyzes this data and produces a report. What we've done in this case over here, as is shown in the red boxes, is we've actually added 27 additional parameters to be showcased on this report. So a user can actually modify this report to immediately and rapidly call out cell counts and frequencies of the functional markers and their expression in particular cell subsets that they might be interested in. For example, we are showcasing expression of OX40, PD-1 on CD4 T cells and its subsets. We can also look at FAST expression on B cells, etc. So this is a highly customizable model where the users can actually modify it to immediately see the parameters that they are interested in. Maxpar Pathsetter also produces what's called progression plots, where we can, within an immune cell type, actually monitor the expression of particular immune cell markers across the cell subsets. So shown over here on the top left, on the top panel, we're actually looking at the CD4 T cell subsets, and we're looking at the expression patterns of these markers that we added. So for example, we can note that the CD7 expression is highest on the naive cells and progressively decreases as we get towards the terminally differentiated effector cells. In the bottom half of that figure, we're actually looking at the myeloid cell populations, and we can actually see that CD33 and CD99 expression, for example, is highest on the classical monocytic population compared to the other non-classical or intermediate monocytic populations. So this is just to highlight that the user can get an immensely good visualization tool and also a quantifiable tool when they're analyzing their data by Maxpar Pathsetter. So to summarize, I'd just like to showcase or tell you that we've added 14 additional antibodies to the base 30 marker panel in the Maxpar Direct Immune Profiling Assay to look at a 44 marker panel. We actually use these 14 additional markers to incorporate 27 additional measurements into the Pathsetter report. Using Maxpar Pathsetter, we can also look at the relative expression levels of each of these markers, as can be visualized in Cen-se plots or even in cell progression plots for a more quantifiable analysis. And scientifically, we also observed data that was already in alignment with what's already been published. For example, increased frequencies of OX40 expression on CD4 T cells in PBMC samples from rheumatoid arthritis samples. Collectively, we've demonstrated the flexibility of this 30-panel Maxpar Direct Immune Profiling Assay to really accommodate expanded additional markers, expanded panels, which will really further facilitate deep immune profiling studies. I'd like to invite you to download the poster at our booth and take a closer look at the data we've shared with you, and to see how the Maxpar Direct Immune Profiling Assay will be applicable to your particular research questions. With that, I would like to thank you for your interest in our work. Thank you.